regulated, should be controlled. This control is done by respiratory system. Means that when the oxygen decreases, what happens? The action of the respiratory system should be increased. The rate of respiration, the depth of the respiration, the ventilation should be increased. Opposite to this, when the oxygen is enough or is more than the physiological range, here we can see that the action of the respiratory system changes. So here I want to talk about two pathways for regulating the respiratory system. The first is neural. Neural is this. You know that the respiration is done automatically. For example, during sleep. During sleep, you have this respiration, and it's done automatically. For this automatically action of respiratory system, we need the internal generator inside of the nervous system, or the neural control or regulation. At first, I want to talk about this part. And the second part of regulation, I want to talk about chemical control. Chemical control means that the oxygen and carbon dioxide and hydrogen ion in fluid body can regulate the action of this respiratory system. So, you know that for respiration, both of inspiration and expiration, you know that respiration includes inspiration and expiration. Before, I repeated this, that you should know that the inspiration is active and expiration is passive. Means that for normal and quiet inspiration, we need diaphragm contraction. The contraction of diaphragm needs two action potential. This action potential is produced inside of the phrenic nerve. You know phrenic nerve. Phrenic nerve is motor neurons for diaphragm and lies inside of the cervical 3, cervical 4, and cervical 5. You know this. Yes. But for action potential production inside of the phrenic, we need a generator before all this. And at first I want to talk about this. So the skeletal, the diaphragm for inspiration needs contraction. This contraction needs action potential. This action potential originates from phrenic. And inside of the phrenic, this action potential originates from another side, inside of the nervous system. Now I want to talk about this source inside of the nervous system. At first, you know that we have two dual or two pathways for respiration. The voluntary pathway, voluntary, and depends on your decision. By your decision, you want to have a high frequency respiration. You can do this. You can change the depth of your respiration. It's voluntary. And it's corticospinal. From the brain cortex, you have decision making inside of the cortex of the brain. And by this decision making, you can have different action inside of your respiratory system. We have no more by this part. The second part of the respiration is very important to us, and is the involuntary, automatically respiration. Is very important to us. Why? Because in sleep we have not any conscious and we have not any decision making, and the respiration is done automatically. And here I want to talk by details about this involuntary or automatic respiration. At first, I want to talk about brain stem. What is the brain stem? In brain stem, we have two important structures. At the upper part, you have pons. At the lower part, you have medulla. Two parts of brainstem are the medulla, upper part and pons, upper and medulla in lower. Okay, so at first I want to talk about different center inside of the medulla. In medulla, you can see two different centers. At the first center is named dorsal respiratory group. Lies in dorsal part of the medulla, and for this is named dorsal respiratory group. The second center inside of the medulla lies in ventral part of the medulla and is named ventral respiratory group. Dorsal respiratory group is abbreviated to DRG, and ventral respiratory group is abbreviated to VRG. At first, I want to talk about DRG and second VRG. But after this, we reach to pons center. In pons center, you can see different center, but one of these centers by the name pneumotoxic center is very important. It's very important to us. By pneumotoxic action, in fact, we have control of rate and depth of the respiration. So the location or the center for regulating or control the rate of the respiration, the depth of the respiration is done inside of the pneumotoxic center. And this pneumotoxic center is located inside of the pons. Dorsal respiratory group. Dorsal respiratory group, when I study on the dorsal respiratory group and activate different neurons inside of the DRG, I see that the activation of each neuron inside of dorsal respiratory group leads to inspiration. And for this, we name this type of neuron as inspiratory neuron. Inspiratory neuron. All neurons inside of the DRG are inspiratory and leads to inspiration. Leads to inspiration. Okay? Opposite to this, when I activate or stimulate the ventral respiratory group, some neuron inside of the VRG leads to inspiration, some leads to expiration. But at first, I want to talk about DRG. In DRG only we have inspiratory neuron. Means that by stimulation of this neuron we have only inspiration. But these neurons have a specific action potential by the name of ramp signal. Ramp signal. Ramp signal. Ramp signal is the name of action potential inside of DRG. DRG. Can you tell me that if you want to select one of these centers as the main center for control and regulation the respiration, if you want to select DRG is main or VRG is main, which of them is main? DRG or DRG?
DRG.
V or D?
D.
Why DRG?
VRG only control the inspiration and expiration during exercise. At the normal DRG.
DRG. If you remember, when I talked about filling balloon, I told that for filling a balloon we need energy. After the expansion of the balloon, we don't give to any energy for empty of the balloon. The expiration is done automatically after the inspiration. So for normal expiration, we don't need any center Because after the end of inspiration, automatically we have expiration. So when I want to select DRG or DRG, I select the DRG because inspiration is important to us. And inside of the DRG only we have inspiratory zone, right? It's enough. Similar to filling or expansion of the balloon. Okay, so, but these center can produce a specific signal or action potential by the name ramp signal. Ramp signal. Ramp signal has this shape. This ramp signal, you can see that for 2 seconds we have this signal. For 2 seconds, we have this signal. During this signal, we have inspiration. But for 3 seconds, this signal is lost. This signal, we have not this signal. And here you can see that here we have expiration. Expiration duration is about 3 seconds. Duration of inspiration is 2 seconds. Each respiration duration is 2 plus 3, 5 seconds. 5 seconds. And each during 1 minute or 60 seconds, we have how many respiratory rates? 12. 12 multiplied by 5 is the same 60. So, but you should know that from the pneumotoxic center, we have the inhibitory signal toward the DRG for 3 seconds. When the inhibitory signal from pneumotoxic reach to DRG, for this time you can see that we have not any ramp signal and we have not inspiration and only we have expiration. So after this, I want to talk about, but this DRG itself can receive input from different parts. For example, from peripheral chemoreceptor, this peripheral chemoreceptor can send its message from the peripheral tissue toward the DRG using 2 important cranial nerves, glossopharyngeal or cranial nerve number 9 and number 10 is the vagus. And baroreceptor. In circulation blood, you read about this baroreceptor. So, but action potential, this signal ramp from DRG goes toward phrenic nerve. At first, should activate the phrenic nerve. By the activation of phrenic nerve, you can see that the new action potential can go toward the diaphragm and can activate the diaphragm. The second center, again you can see this signal ramp here. 2 seconds for inspiration, 3 seconds for expiration. You can see this type of ramp signal. But the ventral respiratory group. In ventral respiratory group, we can see both types of neurons. Means that some neurons can activate inspiration, some neurons can activate expiration. We have both type of inspiratory and expiratory neuron. But VRG has no any role for control and regulation of normal and quiet expiration and inspiration. In normal, quiet inspiration and expiration, the VRG has no any role. But its role is during deep and forceful inspiration and expiration. So, for normal and quiet inspiration and expiration, no need for DRG. In which condition we need deep and forceful inspiration and expiration? We can see that the DRG is activated, and it's specific for deep expiration. For deep expiration, the DRG action is very important to us. Pneumotoxic center. Here we can see pneumotoxic center. Told before that, this pneumotoxic center for 3 seconds can send inhibitory signals toward DRG and can inhibit the signal lamp. This signal. And for this duration, we have only expiration. We have not inspiration. By this, we can tell that the pneumotoxic center is a location for control the rates and depth of the respiration. Means that in normal condition, the pneumotoxic center is inhibitory for 3 seconds, and by this we have 3 second expiration. When the activity of the pneumotoxic center increases, what happens? The duration of expiration decreases, and by this the inspiratory decreases, and by this the rate of the respiration decreases. The depth of the respiration decreases. More activity, you know this, more activity of pneumotoxic less leads to decrease in duration of inspiration, duration of expiration, or it's better to say that duration of the respiration. When the respiration duration decreases, what happens on the respiratory? Respiratory increases. When the inspiration and expiration duration decreases, what happens on tidal volume? Tidal volume decreases because we have not enough time for filling or for empty of the lung. So more activity leads to the less duration for inspiration and expiration, more respiratory rate and less depth. Opposite to this, when the activity of the pneumotoxic center decreases, what happens? The duration of inspiration increases. The duration of expiration increases. The duration of respiration increases. So the rate of respiration decreases. What happens on the tidal volume? The time increases and the tidal volume increases. In addition to this center, we have a specific reflex by the name Henry-Brewer. Henry-Brewer is a reflex that originates from the smooth muscle cells in one of the airways. In one of the airways, we have smooth muscle cells. In this smooth muscle cell, you can see a stretch or mechanical receptor. Means that when the tidal volume increases, by increasing the tidal volume, the stretch on the airway increases and increases, and leads to a stretch on the smooth muscle cell. By this stretch, we can see that the receptor on surface of this muscle cell is activated. By activation of this receptor on the smooth muscle cell, we can see that the DRG center is inhibited. When the tidal volume increases more than 3 times. The normal value for tidal volume is half liter. When the tidal volume increases 3 times more than normal, means that when the tidal volume increases more than 1.5 liter per each inspiration. In this situation, you can see that the stretch receptor inside of the smooth muscle cell is activated and can send an inhibitory signal toward the DRG and can inhibit the DRG. When inhibit the DRG, the depth of the respiration decreases, the tidal volume decreases. And in fact, we can say that the Henry-Brewer reflex, in fact, is a negative feedback. Is negative feedback that can inhibit the DRG. By inhibition of DRG, the tidal volume decreases. When this reflex is activated, when the tidal volume is increased. And we should stop this increasing by this reflex, by the name periglual. The second part of respiratory control is done chemically, and is named chemical control of respiratory system. You know that. When I talk about oxygen and hydrogen and carbon dioxide in fluid body, this oxygen, carbon dioxide, and hydrogen concentration should be controlled, should be regulated by the respiratory system. In fact, we have different type of chemoreceptor. Chemoreceptor means that the receptor can sense the situation of the oxygen, the carbon dioxide, and the ion hydrogen. Means this. We have different types of chemoreceptor, and by this chemoreceptor we can monitor, we can control and regulate the amount of oxygen pressure, amount of carbon dioxide pressure, amount of hydrogen ion concentration. But this chemoreceptor distribution is different. And for this, we divide this chemical control into two parts, peripheral and central control. Peripheral control or regulation is done by chemosensitive receptor inside of the carotid and aorta. Inside of the carotid is a large artery inside of the neck. At the in wall of the carotid, you can see large amount of carotid body. Carotid body, in fact, is the chemoreceptor. Chemoreceptor can sense, can monitor the amount of carbon dioxide, oxygen, and hydrogen ion. And for this, we name this type of chemoreceptor or control peripheral. But another type, you can see at the medulla, very close to the DRG. This is DRG. Very close to DRG, you can see the chemosensitive area. In this chemosensitive area, you have many large amount of chemoreceptor, and for this we name this location for this center as a chemosensitive area. And by monitoring of the hydrogen ion, can control and regulate the activity of DRG. At first, I want to talk about the central, medulla, DRG, and the chemosensitive area. You should know that the main stimulus, the direct stimulus, I repeat this again, the main and direct stimulus for activation of chemosensitive receptor inside of the chemosensitive area is hydrogen. The hydrogen is the main and direct stimulus. Only and only the hydrogen can activate this receptor. But when the hydrogen wants to travel through the brain, through the blood, and goes toward the medulla, at first should pass through the blood-brain barrier. Blood-brain barrier really does not permit, does not allow to pass the hydrogen ion. But it's paradoxic here. The main ion for activation is hydrogen, but we have a barrier that don't allow to pass the hydrogen from blood toward the medulla. You should know that the carbon dioxide easily can pass from this blood-brain barrier and goes toward the medulla. When this carbon dioxide goes toward the medulla, at first reacts with water, and at the end of this can produce hydrogen ions. So Briefly, I want to tell this. This chemosensitive area directly is activated by hydrogen ion, and indirect and indirect is activated by carbon dioxide. Here you can see again the hydrogen from reaction of carbon dioxide with water. But in the peripheral chemolysis, as told before, carotid and the aorta arch, you can see large amount of the chemoreceptor. This chemoreceptor actually, or in fact, is activated by the oxygen. When the oxygen concentration decreases, the oxygen pressure decreases less than 60 millimeter of mercury in arterial blood. When the oxygen pressure inside of the arterial blood decreases less than 60 millimeter of mercury, in this side you can see that the chemoreceptor is activated. But here you can see the carotid body. In this carotid body, you can see two types of cells. Cell type 1, cell type 2, and cell type 2. Cell type 1 or type 1 cell has another name by the name glumus. In fact, the glumus is the same chemoreceptor. The another type of the cell, or type 2, surrounded this type 1 and has not receptor effects, and is a supportive cell. But where I want to compare about is the last slide. You can see the carotid body here, and this is the arterial blood, where the oxygen pressure decreases less than 60 millimeter of mercury. What happened? On the chemoreceptor, you can see a specific potassium channel. This potassium channel depends on the amount of oxygen, means that when the oxygen concentration is very high, leads to opening of potassium channel. When the oxygen concentration decreases, what happens on potassium channel? Potassium channel closes. By closing of potassium channel, the potassium concentration inside of the cell increases and increases. What happens? The positive charge inside of the cell increases and increases, and leads to depolarization. By this depolarization, you can see that the specific calcium channel is activated. Which calcium channel? Depolarization dependent calcium channel, or voltage dependent calcium channel. And by this depolarization, this calcium channel opens. By opening of this calcium channel, the calcium goes inward, goes into the cell. But into the cell, you can see different vesicles. Inside of the vesicle, we have different neurotransmitters, such as ATP. ATP here is neurotransmitter, is not the same adenosine triphosphate. Here is neurotransmitter, ATP. Is different from the ATP as an energy molecule.
So
and
it's an energy molecule?
no, it's not for energy using. It's neurotransmitter here. ATP here is neurotransmitter, such as acetylcholine, such as norepinephrine, such as epinephrine, dopamine, acts here as a neurotransmitter. This vesicle is full of or filled from ATP and acetylcholine and ATP. By calcium release, you can see that here we have exocytosis. By exocytosis, this ATP and acetylcholine released from this glumus cell, from this chemoreceptor, and by activation of its receptor can activate the vagus nerve or the glossopharyngeal nerve. This vagus nerve and glossopharyngeal nerve sends is input to the DRG and can control and regulate the action and the activity of the dorsal respiratory group. Thank you so much. For final exam, what's your idea? What's your opinion? This dive is very
Hard.
high.
They're very hard.
Is enough?
Yeah, they're enough, yeah.
You have problem with reading the book?
No.
It's
Yes.
not that you have a problem, but the exam is only 1 to 2 weeks away, so there's not.
No problem. So the slides is enough for your final exam. And for okay.
Thank you.
Thank you so much.